Today's laboratory activity is Chemistry of Living Organisms, Carbohydrates, Proteins, and Lipids, which begins on page 37 of your lab manual. Please take a look at the objectives that are listed for this activity.
In this lab, we're going to become acquainted with some categories of organic molecules. You might remember from lecture that the categories of biologically important organic molecules are the carbohydrates, proteins, nucleic acids, and lipids. In this lab, we will be dealing with three of these categories: carbohydrates, proteins, and lipids. We'll deal with nucleic acids in another activity.
We're going to start out by looking for some carbohydrates. We're going to look for two kinds of carbohydrates in today's activity. The first one is starch, which is a polysaccharide. So on page 38, you can read about carbohydrates and starch. And the test we're doing, the reagent that we're using to test for starch is iodine. So these are the 5 solutions that we are going to test for the presence of starch using iodine. Now right away you might notice that one of them is starch. So we would expect that one to give us a positive result. And water, of course, is our negative control because water doesn't contain starch, it's just water. So we would expect tube 1 to show us a negative result and tube 3 to show us a positive result. So what I'm going to do is I'm going to follow the instructions in part 1 and then look at the color. So here are the five solutions that we're going to be testing in table 1. I'm going to put 15 drops of each solution into the appropriate test tube, and then I'm going to add 15 drops of iodine, our test reagent, to each tube. Okay, so I've added 15 drops of the appropriate solution to each of these 5 test tubes. Now I'm going to add the iodine. So the 15 drops of iodine have been added to each test tube, and I've placed the test tubes on a piece of white paper so you can see the color better. So you're going to want to record the final color of the solution in each test tube in table 1 on page 39. So here's tube 1, tube 2, tube 3, tube 4, and tube 5. So which tubes are positive for starch? Now we're moving on to testing for a simple sugar. So an example of a simple sugar is glucose, and you can read about that on the bottom of page 39. Our test for simple sugar is described on page 40, and it involves a reagent called Benedict's reagent. Please notice a couple of things about this particular test. One, we can potentially see a range of colors at the end, and the color tells us the relative amount of simple sugar present. The second thing that's worth noting is that this test requires heating. So after we make the test tubes, after we make the solutions, we're going to have to place them in a boiling water bath for 6 minutes. So we're going to set up the test tubes as shown in table 3. So we have the 5 test tubes, each containing 15 drops of the appropriate solution as listed in table 3. Now we're going to add 15 drops of Benedict's reagent to each tube. Now we'll heat each tube for 6 minutes in a boiling water bath. Okay, it's time to carefully remove the test tubes from the hot water bath. Okay, let's take a look at our tubes, and these results go in table 3. So, tube 1, water. Tube 2, glucose. Tube 3, starch. Tube 4, onion juice. Tube 5, potato juice. Please record the colors in table 3, along with your conclusion about the quantity of simple sugar in each tube. Now we're going to test for proteins, our second category of organic molecules. Remember that proteins are polymers of amino acids, and those amino acid monomers are held together with a bond called a peptide bond, and our chemical reagent, Biarritz reagent, tests for the presence of that peptide bond. So we're going to set up the experiment as described on the bottom of page 41 and on page 42. Okay, so we have our 4 test tubes set up, containing water, starch, egg albumin, and gelatin. And we're going to add our Biarritz reagent to each tube. Okay, let's take a look at the color of these tubes. Now this is probably the most subtle test that we do in terms of color change. Let's take a look. So I think you can see the numbers on the tubes as well as the color. So again, here's tube 1. That's water and Biarritz reagent. And I think you can see it's a light blue color. Here's tube 2, starch and Biarritz reagent. Tube 3, egg albumin and Biarritz reagent. And tube 4, gelatin and Biarritz reagent. So you'll want to record the final color of those tubes on page 42 and determine which tubes contain protein and which do not. Now let's take a look at the section on lipids, part 3, which begins in the bottom of page 42. Now we're not really going to test for the presence of lipids. We're just going to explore one of the main characteristics of lipids. So lipids are a very diverse group of molecules. But the thing that all lipids have in common is that they are hydrophobic, which means they separate from water rather than mixing with it. Hydrophobic literally means fear of water. So lipids behave like they're afraid of water. So we're going to look at triglycerides. In this case, we're looking at sesame oil. And we're going to look at the hydrophobic nature of triglycerides and see what we can do to make this oil interact with water. So we have 2 tubes, we're calling them A and B, and we've added 3 milliliters of water to each tube. Now we're going to add 15 drops of sesame oil to each tube. So I've added 15 drops of sesame oil to each tube. I think you can see that the sesame oil is sitting on top because it's hydrophobic. So the water is on the bottom, and the sesame oil, our triglyceride, our lipid, is sitting on the top. And even if I swirl these around, we see the triglyceride stays on top. So I'm going to now add an emulsifier, bile salts, to just one of the tubes. So I'm going to leave tube A alone. I'm going to add 10 drops of bile salt to this tube. Now I'm going to cover both tubes and shake them. Okay, now I've covered the top of both of these tubes and I'm going to shake them vigorously. Okay, I've shaken both tubes vigorously. I'm going to let them sit and we'll come back to them in 2 minutes. So our 2 minutes are up. Let's take a look at these tubes. So here's the first tube. So here's the first tube. It contains just the sesame oil and water. Here's the second tube that contains sesame oil, water, and bile salts, our emulsifier. So please record your observations and the final appearance of the layers on page 44.